Welcome to the WION podcast, where we explore fascinating stories and ideas from various fields. In this episode, we explore a breakthrough discovery, revealing how scientists have uncovered a key process in programmed cell death that prevents cancer from spreading, offering new insight into how our cells self-destruct to protect the body and paving the way for future cancer therapies. What researchers uncovered next could redefine cancer science. In a first, scientists discovered new information about a key process which is involved in the early phases of programmed cell death, also known as apoptosis, which also stops cancer from spreading. The findings of the study, which was led by Dr Luke Clifton of the Science and Technology Facilities Council's ISIS Neutron and muon source in Oxfordshire, were published in the journal Science Advances. The researchers found the molecular mechanism in apoptosis, where cells are no longer needed to undergo self-destruction by activating the so-called death program. Apoptosis is crucial for human life and also plays an important role in biological processes like immune system regulation. It also helps eliminate potentially harmful or cancerous cells from the body, while its disruption can cause cancerous cells to grow and reject treatments. Typically, apoptosis in healthy cells is regulated by two proteins, BAX and BCL2. BAX is a soluble protein which is responsible for getting rid of the old or diseased cells from the body, as it creates pierces through a cell's mitochondrial membrane to form pores which trigger the programmed cell death or apoptosis. Meanwhile, BCL2, which is embedded within the mitochondrial membrane, can offset this process in some cases, which acts to prevent untimely cell death by capturing and sequestering BAX proteins. However, in cancerous cells, BCL2 is overproduced and leads to uninhibited cell growth, and where normal cells stop dividing, cancer cells can keep on proliferating, forming a growing tumor. For the first time, the researchers have found evidence of the involvement of mitochondrial lipids in the cell death process, through a technique known as neutron reflectometry. It enabled scientists to study in real time the way that the BAX interacts with lipids present in the mitochondrial membrane during the initial stages of apoptosis. As far as we can tell, the mechanism by which BAX initiates cell death is previously unseen. Once we know more about the interplay between BAX and BCL2 and how it relates to this mechanism, we'll have a more complete picture of a process that is fundamental to human life, said Dr Clifton, as quoted by the Irish Examiner. The scientists found that when BAX creates pores, it extracts lipids from the mitochondrial membrane to form clusters on the mitochondrial surface, and with the help of time-resolved neutron reflectometry and surface infrared spectroscopy, they were able to see that in two stages. First was the fast absorption of BAX onto the mitochondrial membrane surface, which was followed by a slower formation of membrane-destroying pores and BAX lipid clusters, which occurred simultaneously. Similarly, Professor Gerhard Grobner of Umeå University in Sweden and co-lead of the study said, "The unique findings here will not only have a significant impact in the field of apoptosis research, but will also open gateways for exploring BAX and its relatives as interesting targets in cancer therapy, such as by tuning up their cell-killing potential, as per ANI. Thanks for tuning into this incredible story. If this episode resonated with you, don't forget to hit the like and subscribe button for more captivating stories to come. Stay tuned.